is essentially the evolutionary history of a group. Today, we typically inform phylogeny, or the data that we use to reconstruct phylogeny is primarily genetic data, and at least that's what's most often used to infer relationships, but we can use things like morphology or behavior. And a phylogeny we typically represent with a branching diagram that shows on the x-axis points of divergence, so where species split off to produce more species, and then along the y-axis is time. We can actually calibrate these and make an estimate of the amount of time. On this phylogeny of hominid relationships, starting at primates about the primate ancestor about 7 million years ago, and then moving to humans towards the present, between now and about 1 million or so years ago. Phylogeny, in addition to informing us or giving us a lot of information about evolutionary diversification, it also tells us, we also use it in, or rather it affects your life in a number of ways that you probably weren't aware. It's used in looking at the evolution of disease. One of the best examples of this is HIV, and looking at the origin of that in other primates. Vaccine development, the flu shots that you got recently probably were phylogenetically, or most likely phylogenetically informed. Drug discovery, development of antivenoms, even in forensics, conservation, looking at trying to decide what species we might best conserve. And then even how organisms are classified. When you look at a genus or a family, the species that are in it, typically that's informed through phylogeny. Phylogenetic reconstruction from a computational perspective is not at all trivial. And in fact, the number of possible trees, for us to decide what the best tree is or apply some optimality criterion, we have to search tree space. And the number of trees is dictated by, or it increases with in a greater than exponential manner with the number of species that are on the tree. For example, a tree with only 3 species, there's only one possible tree we could construct. But for 4, it jumps to 3. If we go to 8, there are 10,000 possible solutions. Jump to 10, there's over 10 million. When you get to over 52, there's 10 to the 80th possible tree solutions that have to be explored. So high-performance computing is absolutely necessary to be able to explore tree space and implement an optimality criterion to figure out what tree best fits the data that we've collected. This has been further confounded by the fact that now we can sequence nearly the entire genomes of organisms. It's gotten incredibly cheap. In fact, the cost has fallen off exponentially, over the last 10 years or so. In 2001, it would have cost about $100 million to sequence a human genome. Today, that cost is somewhere around $1,000 or so. And not only can we sequence human genomes for such a small amount of money, we can sequence spider genomes. We can sequence any genome for a really minimal amount of cash. Work in my lab primarily focuses on spiders, so looking at spider diversity and thinking about the evolution of this incredibly diverse group. In fact, it's the largest group of animals known that is exclusively predatory. Spiders make their, all spiders make their living killing things and eating them. And the way that they do this is through their incredibly innovative use of spider silk. All spiders, or rather the defining characteristic of spiders, is their ability to produce silk from these abdominally placed appendages termed spinnerets. Silk is used in nearly every stage of the life history of a spider, everything from building egg sacs to protecting themselves, protecting their young in reproduction, and in prey capture. And in fact, silk is an incredibly strong natural substance. Silk has a tensile strength that exceeds steel, and it also has an extensibility that's far greater than steel or man-made sorts of products like Kevlar. If you could imagine a pencil-thick a pencil-thick strand of spider silk could actually stop a Boeing 747. It's an incredibly strong, natural-made material. And work in my lab is focused on looking at the evolution of spider silk. This is a rudimentary or a very traditional view of spider phylogeny, so the evolutionary history of spiders. We've got trapdoor spiders and tarantulas down here, and then you move up into more derived spiders. But the spiders that we're really interested in are the ones that build orb webs. So this everybody's probably seen an orb web. It's the big webs that spiders build on your porch, or they build across a trail. You tend to walk into them and get them across your face. But these have long been considered by spider biologists to be a sort of it's kind of a natural engineering marvel. You know, you've got to imagine that this web has to, they capture flying insects with these, oftentimes heavy insects. The insect slams into the web absorbs the force of the impact. The web is sticky, so it holds the insect, or it holds the prey item long enough for the spider to scurry down and envenomate it. It's a very diverse group, and there are two major groups within this, the dinopoids and the araneoids. The dinopoids are considered to be the primitive orb weavers, and the araneoids and their relatives are the more advanced. What separates them is what makes their webs sticky. So dinopoids make this fuzzy Velcro-like adhesive threads, whereas araneoids use gluey adhesive droplets on their webs. And these are way more stickier and thought to be produced at a much smaller economic cost. Spider biologists for many years had considered the primitive orb-weaving spiders to be unrelated to the more advanced, the adhesive orb-weaving spiders, that they didn't share a common ancestor on the spider tree of life. The implications of that is that spiders would have had to have come up with this sort of remarkable engineering feat more than once, at least 2 times on the spider tree of life. But in the 1980s, looking at physical characteristics and behavior, spider biologists all sort of decided at once that no, that web could have only evolved one time, and that all the spiders that build orb webs share a common ancestor. And a lot of the work in my lab has been focused on applying genomics and next-generation sequencing technologies to reconstruct the spider tree of life and address that question once and for all. How many times, did the orb web evolve once, or did it evolve or did it evolve multiple times? So we use Illumina-based sequencing, a number of high-performance computing approaches to assemble the data, build DNA data matrices, and then reconstruct spider evolutionary history, or infer an evolutionary history for spiders. The work that we've been doing was actually featured in the work from my lab and a couple of other labs around the country was actually just featured in the October issue of Science, where essentially these data and these approaches that we've been using have almost overnight transformed our view of spider evolution, both in terms of their webs and their venoms and a number of other really cool things that spiders do. I'll cut to the chase here and say that we've now sequenced large snapshots of many spider genomes and have reconstructed their evolutionary history. And the end, sort of the punchline on this, is that the primitive orb-weaving spiders are not related at all, or at least very distantly related, to the adhesive orb-weaving spiders. So that actually takes us back, flips the whole situation back on its head, and takes us back to the 1980s, where the primitive orb-weaving spiders and the more derived adhesive orb-weaving spiders don't share a common ancestor. The interesting thing about this is that what that means is the orb web either evolved multiple times or it had to be lost multiple times. And using computational approaches, we've reconstructed the evolution of web type across the spider tree of life. And what we found, represented in yellow here, is what we call the ancient origin of the orb web. And that is that spiders gained the orb web a long time ago. I'll talk about that in just a second, and then lost it multiple times. And in fact, there's a huge loss up here, things like wolf spiders and jumping spiders. Most of you have heard of those. So a bunch of spiders lost it, lost a couple of other points, but actually the origin of the orb web goes way back in time. In fact, it goes back about 213 million years ago, which is the early Triassic. So using molecular dating approaches and fossils as calibrations, we've reconstructed the origin of this to the late Triassic. What that means is that at this node, so again, this is on the tree of life, or on the spider tree of life, where we've got jumping spiders and wolf spiders. What we found is, again using computational approaches, is that the largest amount of spider diversity, if we look across this tree, the most rapid diversification rates, and this is computed using Bayesian mixed models, the most rapid diversification rates are when spiders actually lose that orb web. So getting rid of the orb web turned out to be a great sort of innovation in the spider history of life. And in fact, that occurred, we dated that at about 138 million years ago, which is during the early Cretaceous. During the early Cretaceous was what we termed the Cretaceous terrestrial revolution, and that's when there were some major changes in the plant and particularly insect life that was going on at the time. So taking the orb web and punting it, as it turns out, was probably a major innovation for spiders and spurred a lot of diversity during the early Cretaceous. So I'll leave you with just a few things. One is I hope in these brief few minutes, I've convinced you that spiders are at least are a relatively interesting group of animals, and they should be feared as much as they are. We've had some major changes in how we view the spider tree of life, fundamentally changing what we know about evolutionary pattern and process, and that genomics and big data and HPC have been the only way that we've been able to address some of these questions. So with that, if there's time, I'm happy to take any questions. Thanks.
Actually, Dr. Jason Bond, everyone.
Thanks.
And will you stick around for questions?
Sure.
We're going to do that after the wrap-up. Okay.